We're going to go ahead and get started. It's a little after 10:30. We're so grateful for all of you that came out on August Saturday to attend this event. I'm really excited about this event. I've been trying to get Dr. Goldman to come talk for a while, and now we're making it happen, so it's great. It's especially great to see so many of you in the summer. I know a lot of you have travel plans. So today's talk, as you can see, is called Parkinson's in the Environment, Causes and Prevention. And we're pleased to welcome Dr. Sam Goldman from UCSF. The talk is going to provide an overview of what we currently know about environmental epidemiology of Parkinson's and review some of the known chemicals, particularly TCE, trichloroethylene, which is an industrial solvent. He's going to talk about a study that he did with Dr. Carly Tanner, and many of you know Dr. Tanner from UCSF. It's called the Camp Lejeune study, and it's about a marine base in North Carolina where there was a known TCE exposure. So it's a very unique study because there's a known exposure and they didn't have to infer exposure based on self-report. So they compared Camp Lejeune with another marine base called Camp Pendleton. So we'll hear all about that. I'm particularly excited about that because my background is also environmental epidemiology, so I'm really interested. And we're also going to hear about some ways that we can get involved if we're interested in reducing some of our exposures to some of these environmental contaminants. If there's any people in the audience, I know we do have one person, Renee, who has joined us. We're so grateful, who was stationed at Camp Lejeune in the 70s and 80s. If there's anybody else, we'd love to hear from you as we engage in the discussion at the end. We'll have about a 45-minute talk, and we'll have questions and answers at the end. Again, my name is Monique Buffler. I'm on the board. I'm on the events committee with Todd over here working the camera, and Hari in the back. Thank you so much, Hari. And also our beloved Kathy. I'd like to invite Kathy to come up front. Kathy has been, come up front. As many of you know, Kathy has been instrumental on the events committee as well as the programs committee at PediActive. And Kathy has just taken another opportunity at the Chinese immersion school. And so we'll be leading PediActive. So we just wanted to honor her and thank her for her 5 years of service. It's an amazing asset for us. I mean, with her generosity and her spirit and her kindness. And so we really love working with you, Kathy. And sad to see you go, but really happy for you. Thank you. You want to say anything?
I love you guys so much, and I'm so sad to go. Thank you so much for everything. It's been a wonderful 5 years, and I'm so sad to go. And I'm not going anywhere. I'll be in Oakland, and I'm going to volunteer, and I'll be right around the corner. So call me anytime.
All right. So just a quick few housekeeping things. The restrooms are right back there where it says exit. And there's also some coffee and treats on the back table. If you haven't already, please stop by the front so you get a name tag and sign up. So just a reminder, we are recording. That's what Todd's doing over there. We'll post on our YouTube channel. So I just wanted to let you know we won't record the Q&A, probably just the talk. So I'll go ahead and introduce Dr. Goldman. We're really excited to have him with us today. He's a local. He's a researcher and professor at UCSF and the San Francisco VA. Also did some graduate work here at UC Berkeley, go Bears. He got his undergraduate in neuroscience at the University of Michigan, and then did his medical degree at the University of Texas in Houston. And he's published extensively on the environmental epidemiology of Parkinson's disease with a focus on risk factors, environmental risk factors of pesticides, solvents, and head injuries, and their interaction with genetic susceptibility. In addition to his ongoing research as an emeriti professor, he has begun an intensive study of foods, wines, and lifestyles of southern Spain. And we also just learned that he's a talented musician. So I think we're ready to hand it off to Dr. Goldman. Thank you.
Thanks, Monique. Thanks for inviting me. Thanks for everyone for coming today. And I hope this will be interesting to you all. I know you're a super highly educated group and know a lot about Parkinson's disease in many respects, a lot more than I do. I should say I'm not a Parkinson's clinician. My clinical life was in environmental medicine, so I'm not very knowledgeable about many of the clinical trials and the treatments, the newest treatments. But if you have any questions as we go through this, I'm happy to stop and answer them. Here we go. Ready?
Yes.
All right, so just very quickly, the epidemiology of Parkinson's disease, so who gets it, where do they get it? Are there differences geographically? Are there differences between sexes? How long has it been around? How many people get it? It's the descriptive epidemiology. It's the most common cause of Parkinsonism. There are lots of other causes of some of the clinical features. And we think that Parkinson's disease has been around for a very long time. There are descriptions that seem similar to Parkinson's disease in some writings of Maimonides 1,000 years ago, and also in some ancient Indian texts. But the average lifespan was, 35 years. So there wasn't a lot of Parkinson's disease for sure. But it wasn't until 1817 that James Parkinson described the syndrome completely. And he predominantly described it from his window, looking down on the street in London. He only examined, I believe, 2 patients. And Parkinson's, currently, we think it affects approximately 1 million people in the US. Its prevalence is about 2% in persons over age 65. And as you can see from this study, which is from Kaiser, here in the Bay Area, Northern California at Kaiser. It's quite rare, as you know, under age 50, but it's there. And then it takes off exponentially in terms of its frequency. And at all ages, men are more frequently affected than women. We don't know why that is, but it's very consistent across all populations. And we think that whites are more frequently affected than blacks, although there are certainly issues of access to health care and ascertainment of disease frequency in underserved populations. The global burden of Parkinson's disease, this is from a number of years ago now, a paper by Ray Dorsey, who some of you may be familiar with his book. This shows where Parkinson's rates are, not rates, where Parkinson's occurrence is increasing most rapidly. And China, Brazil, India are the areas where there was predicted to be, it's kind of blurry on the screen, but it's predicted to be 100% increase from 2005 to 2030. And this is the global estimate of how much Parkinson's disease there is in the world. And this shows that in 2040, we estimate that there will be over 17 million people worldwide with Parkinson's. So I'm going to just jump into some of the causes. This isn't going to be thorough, but it's a lot of information nonetheless. I'm going to talk mainly about pesticides and solvents. But the debate as to whether Parkinson's is predominantly genetic or environmental goes way back. Charcot wrote that it's not a familial disease, but Gowers thought that many people did have a family history of Parkinson's, which would suggest a genetic basis. And this is super unreadable, but what this shows, on the upper right, so we have learned a lot. I got into this field of research in the early 90s when I met Carly Tanner, who was teaching my tox class at Berkeley when I was getting my MPH. And we knew nothing about Parkinson's disease 30 years ago. So the progress has been incredible, though not fast enough. But in the 90s, we discovered this large family. It was an Italian-American family called the Contursi family. And Larry Golby, who was at Rutgers, did this initial research, and he put together this pedigree. So this is a large family, and anywhere, it's hard to see, but anywhere where there's a dark box is someone with Parkinson's disease in this family. And it was inherited in an autosomal dominant fashion. So 50% chance that if a parent had it, that the kid had it. And they figured out what the gene was that was being transferred in this family, and it was the alpha-synuclein gene. Many of you may be familiar with that word, that term. And it was a single mutation, one base pair mutation, the A53T mutation in the alpha-synuclein gene. And this set off a huge search, right, across the entire world of Parkinson's research to try to see, okay, does everybody with Parkinson's disease have this mutation? And it turns out, no, virtually no one does, except for the Contorsi family. And but what this discovery did lead to was the identification of alpha-synuclein, the protein made by this gene, as the major constituent of the Lewy body, which Lewy bodies are the intracellular inclusions that we see in pathology in neurons, particularly dopaminergic neurons. And so this was a huge breakthrough, that we identified this protein, that even though people don't have this mutation, they do have this aggregation of protein in their brains with Parkinson's disease and also elsewhere in the body. So after this discovery, about 10 other genes were identified over the next 30 years. And some of you may be familiar with the PD Generation project. So this is a giant, mostly North American, but worldwide effort to truly figure out what proportion of people with Parkinson's disease have some genetic basis or genetic predisposition. And what this has led to, these pies show, on the left, the lighter colored slice is the proportion of people in PD Generation who have a variant in any of these 10 or so genes that have been linked with Parkinson's. And it's about 13%. And then they break it down on the right to which particular genes they are. There's one called GBA, one called LARC2, and those two are the most common. But they're not that common. So, okay, so 13%. So what's the other 87%? And this, it's kind of complicated looking, but it's not. On the bottom is the frequency of a genetic variant, and on the y-axis is the risk associated with that variant, the risk of Parkinson's. And what you can see is that there's a handful of rare variants on the left at the top that impart a large risk. But down at the bottom right, there are all of these, there are about 90 now, variants in genes that impart a very small increased risk, like maybe 5 or 10% increased risk. And so in total, all of these, what we call highly penetrant or Mendelian variants, and these common single nucleotide polymorphisms or SNPs that impart a very small risk, in total, we think they account for about 25% of Parkinson's. So, this is the increased incidence of Parkinson's, and it is not related to age. This is adjusted for age. So we know there's more Parkinson's because people are living longer. So, but when you take age out of it, we still think that Parkinson's incidence is increasing at about 10 to 25% per decade over the past 50 years. Well, that doesn't sound like a genetic disorder, does it? We conducted this twin study, Carly Tanner and I worked on this 30 years ago. Twins are a great natural experiment for trying to tease apart whether something is environmental or genetic. Identical twins are genetically identical. So if one twin has Parkinson's, if it's entirely a genetic disorder, the other twin will always have Parkinson's, right? And we compared them, and that's called concordance. When one has it and the other has it, they're concordant. And we compared the rate of concordance in identical twins versus fraternal twins. Fraternal twins are only, they're just like regular brothers or sisters, they're 50% the same genetically. So if a disorder is predominantly genetic, the concordance rate should be much higher in identical compared to fraternal twins. And what we found was that if onset of Parkinson's was before age 50, there was a very strong genetic component. It was 80% here. But if onset was after age 50, there was only 20% concordance. And that's very similar to what I just put up in the prior slide, that we think the total genetic contributions are around 25%. So we concluded from the twin study that environment's a major contributor to disease risk. So that's not Parkinson's disease. That's MPTV. Some of you may be familiar with the book, The Case of the Frozen Addicts, written by Bill Langston. This was a group of people who all started presenting to emergency rooms in the 1980s around Silicon Valley. And Dr. Langston was a young neurology resident at the time, and he recognized these people were all coming in with acutely severe Parkinson's disease that was responsive to levodopa. And they figured out they were all sort of part of a IV drug use club, where they have a chemist synthesize drugs that they thought would get them high. This was actually a Demerol derivative. And they found this chemical, they isolated it, and this really launched a whole new search for environmental causes of Parkinson's disease, because it's this thing out there in the environment. Nobody knew what this chemical was. And so it started a search for similar compounds in the environment that might be causing Parkinson's disease. Parkinson's is really hard to study. There's a huge misdiagnosis rate. Many of you with Parkinson's maybe were misdiagnosed at some point in your lives. It's very different from person to person clinically, symptoms. We think that the underlying pathology and the underlying causes are probably very different person to person. And it's also a disease of aging, and so it's really hard to know if we're studying exposures, what people might have been exposed to 20, 30, 40, 50 years ago. That is really hard to measure. But we've measured some. Every line on this is a study of pesticides and Parkinson's disease from around the world. Lots of different study designs, lots of different countries. It's hard to see, but the kind of the solid line, slightly to the left of center, that is 1. So anything to the right of that means that the study found an increased risk of Parkinson's associated with pesticides. And overall, I think it's about, it's not so good. Overall, they find about a 60% increased risk. The problem with this work is that it's very rarely have any specific pesticides been studied. It's just sort of general information about pesticide exposure. About 20 years ago, we started this study in the agricultural health study, which was put together as a large cohort, 84,000 professional pesticide applicators. So those people know what they were exposed to because they had to keep records for the government. And this was in 2 states, Iowa and North Carolina, and we tried to find everyone in this population with Parkinson's disease, and then did what's called a case control study. We compared exposures in people with Parkinson's to exposures in people without Parkinson's. And so in total we had 110 people with Parkinson's and 358 control people. We asked them about 31 specific pesticides, and only 2 were found to be significantly associated. And those 2 were paraquat and rotenone. And individuals who had used those physically, mixed them or applied them to crops, had about a 3-fold increased risk of Parkinson's. Big effect. And that's, you can buy rotenone on Amazon if you're interested. And rotenone is actually considered an organic pesticide, and until about 10 years ago, it could actually be used on organic produce because it's derived from a plant. So what's amazing about that finding is that rotenone and paraquat both produce an animal model of Parkinson's in rodents. They produce the same pathology, they produce a movement disorder. They are all like MPTP, which I showed you the effects of that. They are mitochondrial poisons, they cause oxidative stress, and you can see the chemical structure at the bottom, that's MPTP on the left, and that's paraquat on the right. Very similar chemical structure. This is paraquat use from 1992 to 2012. You can see that's California, the Central Valley, very, very dark. Paraquat is banned in most countries on the planet. Not here, not in the US, and not in a few countries in Central America. It's banned pretty much everywhere else, but usage is actually increasing in the United States because glyphosate usage, is another herbicide, is lessening. So they're replacing it with paraquat. This is another more recent study from a research group at UCLA. This shows, you can't really see it, but basically anything on the right that's above that, that red horizontal line is significantly associated. Those are all pesticides, lots of different pesticides. So now we are learning more about specific pesticides and associations with Parkinson's. This map on the left is in the Fresno area and shows the usage of these compounds still. And this is on a global level, this is looking at change in the amounts of pesticides by weight internationally. If you recall that figure that I showed, you can see some overlap, but the areas where it's really increasing the most are in Canada, South America, and Russia, and some areas in Africa. It's actually decreasing in much of Europe. I'm going to switch gears now and talk about trichloroethylene. TCE is what I'll be calling it. So there have been a lot of studies looking at solvents in general, epidemiologic studies, and for the most part, they're not very good. And that, again, is a limitation of people don't know what they were exposed to 30 years ago. We know that acute solvent intoxication, so people might deliberately or accidentally drink some gasoline, for example, that can cause Parkinsonism, but it's very different from Parkinson's disease. They have damage to their brain very diffusely, and they might have some symptoms that are similar to Parkinson's, but it's not Parkinson's. It's not a model of Parkinson's disease. The solvent associations with Parkinson's have historically been very inconsistent, but as usual in epi, specific compounds are rarely studied. So back in 2008, there was a report by Gash and colleagues from Kentucky, and he was a PD clinician, and he recognized that several of his patients all worked in the same little industrial plant. And he went out to this plant and he examined some of the co-workers of the people with Parkinson's and found that they had some mild Parkinsonian signs, even though they hadn't been aware of it or hadn't been diagnosed with anything. And what was remarkable about this little work site was that they all worked around what's called a vapor degreasing vat that was full of trichloroethylene, TCE. And these are commonly used in all sorts of light industry to degrease things, degrease metal parts, degrease plastic parts. Basically, this tub down at the bottom has got liquid TCE. As a heating coil down at the bottom, it heats it, becomes a vapor, the TCE goes up. You dip your stuff down in a basket and it cleans it beautifully, it works great. TCE gets up to the top, there's a cooling coil, it condenses back out and drops back down into liquid form. a lot of it escapes into the room. And these guys have worked with it for 20 years or more. So we thought this is really disturbing and needs to be followed up on. So we had already collected occupational histories in our twin study, and we tested this hypothesis about exposure to TCE and other solvents in the twin study. The way we did it was by comparing discordant pairs, so pairs where one twin had PD, one didn't. We had 99 pairs like that. And we estimated whether they had been exposed to TCE and other solvents, and this is what we found. There was a slightly increased risk for exposure to things like hexane, toluene, and xylene. There was a 6-fold increased risk for people who had worked with TCE, and a 10-fold increased risk for people who had worked with PCE, which is a very similar compound as PERC or tetrachloroethylene. So, really big effect sizes. And this is what TCE does in animal model. On the left is the control animal, and this is a stain for dopamine neurons in the brain. On the left is a control, they just got saline. On the right, they got TCE. And you can see the drastic loss in the number of neurons in the animal that received the TCE. And we think we know the mechanism of toxicity, though maybe there are others, but TCE gets converted into this compound called TACLO, and we know that it forms spontaneously in rats who've been exposed to TCE. And again, look at the look at the structure of TCE compared to MPTP. Very similar. And like MPTP, and like retinones, and like paraquat, TCE is a mitochondrial poison. So, I'm going into Camp Lejeune now, which was supposed to be the entire talk I'm giving today, but I've been diverted here. So what is Camp Lejeune? Camp Lejeune is one of the largest Marine Corps bases in the country. About 1/3 of all Marines rotate through Camp Lejeune at some point. It's in North Carolina. It's been there for a long time. The history that we're going to focus on goes back to 1953, when we think the wells on the base became contaminated with both TCE and PCE, that other solvent that also seems to increase risk. They were contaminated from a machine base or machine shop on the base, and also a dry cleaning shop that was off the base, and these are both used as dry cleaning solvents. The contaminants weren't discovered until the 1980s, when the Clean Water Act was passed. In the late 70s, they finally actually had to test wells for water purity. And they discovered the contaminants, they closed the wells, and it became an EPA Superfund site. The levels from 1975 to 1985, the maximum EPA allowable level is 5 micrograms per liter. TCE was 366, and PCE was 85. There were over 1 million veterans and civilian workers also who were exposed. They could have been exposed lots of different ways, right? So drinking it, cooking with it, you can inhale it. Bathing, you can inhale it and get absorption through your skin. And recreation. So in 2017, the VA determined that people who had been at Lejeune between 1953 and 1987 were eligible for benefits if they had Parkinson's disease. And the families were eligible for health care coverage. But at this point that determination was made, the total epidemiology, so our twin study, the GASH study that I showed you, there were only 16 cases of individuals who had PD who were exposed to TCE. So we felt that's not enough information. We need to be more certain about this relationship. So we, luckily, were funded by the VA to conduct this study. And what we wanted to test was whether Parkinson's risk was higher in veterans who had served at Camp Lejeune than veterans who had served at another Marine Corps base, Camp Pendleton, out in California, who were presumably not exposed to TCE. And we also wanted to test some of the prodromal features, so other symptoms that often precede Parkinson's diagnosis. So we took advantage of a cohort that the ATSDR, which I I don't even know if it exists anymore. As of a few months ago, when it was part of the CDC. I'm not sure the CDC exists anymore, but at any rate, they existed then, and we were able to put this cohort together of individuals, 340,000 vets who had served at either Lejeune or Pendleton during these 10 years, and they had estimates of exposure levels to these compounds in the water. So we searched for people in the VA healthcare system, in Medicare, in diagnosis of Parkinson's, and then I reviewed their medical records, over 1,000 medical records that I reviewed, and to confirm the diagnosis was correct. Only 46% of people in the VA healthcare system with PD diagnosis had PD. So that just shows you how difficult it is to do this kind of research, because there's so much misdiagnosis. They were relatively young, but what's most interesting down at the bottom is that they were on the base an average of about 2 years, so relatively brief exposure period. These are our results. We found a total of 430 people at both bases, way more at Lejeune, and we estimated about a 70% increased risk of Parkinson's disease for veterans who had been at Lejeune compared with those who had been at Pendleton. The latency from exposure until diagnosis was 34 years. We also found increased risk of having a prodromal feature. So the cohort is still quite young. Their median age. Prodromal means preceding. It simply means preceding the diagnosis. And as many of you are aware, and I'll show you a slide, Parkinson's develops over a really long period of time. We think that the disease process, I compare it to coronary artery disease. So people might have high blood pressure or might have diabetes for 30 years before we recognize that they have coronary artery disease. Same thing with Parkinson's disease. We think that the underlying process is at least 10 years, and it's probably more like 30 or 40 years before people are diagnosed with Parkinson's. So prodromal features are reflective of this underlying process that precedes the formal diagnosis, right? The frank motor symptoms that usually lead to people being diagnosed. So things like subtle tremor, loss of sense of smell, certain skin rash, seborrheic dermatitis, these other features. So we also found that those were significantly more common in Camp Lejeune. We also tested whether people who were exposed at Lejeune might have a different disease course, perhaps a more severe progression. And we were able to, I'll skip through this, but we were able to, about 2/3 of the people who were at Lejeune were thought to be exposed, and 1/3 were thought not to have been exposed. This is a very coarse estimation. And then we looked at the time until certain outcomes that we were able to easily extract from the medical record. And the red line is people who were exposed. And on the left is time until they had a diagnosis of psychosis, much more rapid in the people who were exposed. The upper right is time until their first fracture, which is of course related to falling. Much quicker time to a fracture in people who had been exposed. And on the bottom right is the time until a fall. And that's really the clearest of them all. You can see that people who had been exposed to TCE were more likely to fall sooner. So not only does TCE seem to increase risk, it also seems to increase the rate of progression. And this shows what we call a dose response, which makes us really confident in the observation. So people who were unexposed are the comparison group on the left. We call that a risk of 1. People who were exposed to a low amount are in the middle, and people who were exposed to a high amount are on the right. And you can see what we call a dose response for each of these 3 severity outcomes. So Most of you weren't at Lejeune, so does TCE matter? Well, TCE was in everything. It was in all sorts of home products, carpet cleaners, spot removers, whiteout, you name it.
Decaffeinated coffee?
It was used to decaffeinate coffee until the late 70s. It was used as an anesthetic, particularly during childbirth, until the late 70s and even later in the UK. So it was in everything. And not surprisingly, it got into the water, not just at Lejeune. So it's the most frequently reported organic contaminant in US water supplies. And there are these so-called plumes that get into, once it gets into the soil, it sticks around. If it's in the air, it breaks down, it goes up into the atmosphere and it's gone. But if it gets into the soil, it persists. So this is a plume. These things get into the soil and the groundwater, and they travel slowly over time. There's one in Tucson, one in Dayton, Ohio, one in Brooklyn, Minneapolis, and Mountain View. Mountain View actually has the most EPA Superfund sites, or Santa Clara County, of any county in the US. And it's contaminated, yes, but the reason for that is because they had a very progressive, active mayor who tested. He was worried about this, so he looked, and if you look, you will find this stuff. This is nationally, this is TCE in public water supplies. The darker circles and the bigger circles are more concerning. And this is how most of us get exposed to TCE. It's through vapor intrusion. So these plumes, you live where maybe there was a dry cleaning plant 30 years ago, people don't even know it was there, it's gone now, and they put a school over it, or built some new houses. And it can get into the house and actually concentrate in the house. It gets in through around pipes where they enter, and through cracks in the foundation, like radon, for those of you who remember that, or from the Midwest. So that's how most of us currently get our TCE exposures. All right, so just to review what we've gone over, MPTP is mitochondrial poison. Rotenone is a mitochondrial poison. Paraquat, and TCE and PCE. There are lots of other possible mechanisms of toxicity, but we know they overlap at the mitochondrion, which is the energy producer in all of our cells. I'm going to switch gears again, just talk very briefly about air pollution. The data on air pollution and Parkinson's disease is not super strong, but it's really hard to study. And there are some reasons to suspect that it's highly relevant. Particularly small particles, nanoparticles, you may be familiar with that term, we can barely even measure them. They're really hard to measure, but they are produced largely from cars, combustion. Diesel's particularly good at producing them. And they are tiny little particles that can just go straight through the nose into the brain. And so not only is it the particle itself that we are concerned about, but the particles can carry other things that get stuck onto them. They're really good delivery vehicles to getting into the brain. And just looking at air pollution, so on the left is the chart I put up before, showing the rates of Parkinson's disease, where they're increasing the fastest, and on the right is levels of air pollution globally. You can see they're highest in India, China, and more recently in the parts of Africa that are developing. And this is a study, these are electron microscope photographs, basically, little metallic particles. These are from autopsies of human brains of people who lived in very polluted cities, Manchester, England, and Mexico City. So these are nanoparticles, we believe, that got in from traffic pollution and got into the brain through the nose. So even though the data aren't super strong yet about this, we think that air pollution is a real significant contributor to Parkinson's risk. So, to summarize, what increases the risk of Parkinson's? I didn't talk about a lot of these, but we think that, especially the things that are bolded on the left, increase the risk. So head injury causes inflammation, can make the brain leakier, allow more bad stuff to get into it. Pesticides are discussed somewhat, some solvents. Rural residents has been long observed association, and that is probably related to pesticides. Diabetes is increasingly being recognized as a risk factor for Parkinson's and PTSD as well. Lesser on the left, things like certain occupations. So not the occupations you would expect. So the occupations most strongly associated with Parkinson's are doctors, lawyers, clergy, not farmers and people working with solids, but again, that may just reflect our inability to conduct these studies very rigorously with regard to exposure assessment. And reduced risks. Many of you probably know that there's a reduced risk has been observed with cigarette smoking. There's a lot of, that's absolutely certain. It's observed in every study that's ever been conducted that people who smoke have a lower risk of Parkinson's. That doesn't mean that smoking prevents Parkinson's. It may be, and this is my personal bias, I think that people who are going to get Parkinson's don't like to smoke as much. I don't think it's the smoking that affects the Parkinson's, I think it's the Parkinson's that affects the smoking. I think they don't get the buzz, whatever it is that makes people smoke. There's also a very consistent association with coffee drinking, with caffeine consumption. And now there's some dietary associations. Animal fats seem to increase risk, and Mediterranean diet may decrease risk. Physical activity is likely protective, and there are several studies that non-steroidal anti-inflammatory medications may also be protective. Okay, so how am I doing on time?
Question.
Okay, so these were epi studies, right? Epidemiology studies were studies in populations. And I was talking to Todd earlier. Everyone's Parkinson's disease is different. You're not a population. You're an individual. And we measure these things in populations, but everyone's cause is different. And these are called causal pies. I'd like to show this example. It's simple, but I think it's important to remember. So A and B are component causes. When that pie fills up, you get the disease, whatever it might be. So here's an example, right? This disease is a broken wrist, and what caused this person's broken wrist was they were climbing stairs, it was icy, they had osteoporosis, and the porch light was burned out, right? Without one of those, they wouldn't have broken their wrist. This other person, stairs, ice, high heels, and alcohol. Take away one of those, and they wouldn't have broken their wrist. And I think the same thing is with Parkinson's disease. So this individual, head injury, TC, pesticides, genetics, diabetes. This individual, a different causal picture. This individual doesn't have Parkinson's disease. They had some exposure, but altogether, their genetic risk, their environmental risk added up is not enough to lead to the illness. So here's an example, one example of what we call gene-environment interaction. So this is a combination of a genetic risk factor and an environmental risk factor. In this case, the environmental risk factor is paraquat, the herbicide, and the gene, I haven't talked about genes much. This is GST-T1. It's a common metabolic gene that detoxifies things in the body. And that's why we looked at these two in combination. So it's an antioxidant metabolic enzyme. We know that glutathione levels, which this gene is related to, are low in people with Parkinson's, and 20% of the population doesn't have a functional GSDT1 gene. They just don't make it. They don't make this protein. So we have 4 groups that we're looking at here. The first column on the left, GSDT1 present. So these people they have a functional gene, they make this protein, this detoxifying protein, and they weren't exposed to paraquat. That's our comparison group. The next column to that, GSDT1 null, paraquat no. So those people don't make the gene, or don't make the protein, they don't have a functional gene, but they weren't exposed to paraquat. The next group, they have a functional gene, but were exposed to paraquat, and the last group, they don't have a functional gene and were exposed to paraquat. So that's our reference category. If you just have a non-functional gene, very minimal increased risk. If you were exposed to paraquat, but your GSDT1 gene works, 50% increased risk. If you didn't have a functional gene and were exposed to paraquat, 1100% risk. And essentially, in this study that we conducted, basically everyone who was exposed to paraquat and didn't have a functional GSDT1 gene had Parkinson's disease. This is an extreme example of what we call gene-environment interaction. And those are ways of measuring the impact of that interaction. But this is one example, and I think this is the rule, right? You know, very few people get enough paraquat exposure so that they're all going to get Parkinson's. They have to have some other exposure or some genetic factor that interacts with those exposures. I'm going to throw this in near the end here, that I think, and I don't have a lot to base this on, but it is my belief after 30 years in this field, that I think that Parkinson's, the most important environmental exposures are the early ones. And I'm going to give you a little bit of reason why. So I mentioned about the prodrome. So, kind of hard to see, but the red box, this is a point in time on the bottom, on the x-axis, as we move across time in someone's life. The diagnosis of Parkinson's disease is that red box. And then the symptoms of Parkinson's disease that we're all familiar with. But to the left of that is what we call the prodromal period, and where things like constipation or loss of sense of smell. We know that that occurs years to decades before. So whatever is causing Parkinson's is also occurring years to decades before. I want to go back to this twin study. At the bottom, what we noticed in this was that the concordance in the fraternal twins, so fraternal twins are just like regular brothers and sisters in terms of their genetic similarity. We found a 13% concordance in fraternal twins. And in all the studies that have been done to date, looking at the risk factor, if you have, what's the risk that your brother or sister is going to have Parkinson's? In all of those studies, it's about 4%. It's not very high. But we found 13% in twins. So what's different about fraternal twins from regular siblings? What's different is they shared a uterus, and they shared an early life really closely. So this difference, 13% versus 4%, to me, really implicates early life exposures. Young brains are sensitive, but their bodies have very large surface area to volume ratio. Their livers don't metabolize things as well, and their brains are leakier. They let stuff in. I'm going to talk very briefly about ways that the immune system might get sensitized early on to increase the toxic potential of things that people are exposed to later. And then there's this thing called epigenomics. I'm not going to talk about epigenomics, but basically it's a way that DNA gets modified by environmental things, and it affects the way, it doesn't change your genes, but it changes how your genes get turned into proteins. And these a great example is these mice here. The mouse on the left is the, quote, normal mouse. They're identical twins, genetically identical. But the mouse on the right, its mother got a folate supplement during pregnancy. The mouse on the left, it didn't. So it totally changed the adult mouse based on what the mother was exposed to. And that, I think, is the model here. I'm just going to show a couple of examples here. So neonatal lead exposure. I'm talking about Alzheimer's disease, but there are definite, aspects of Alzheimer's and Parkinson's that we think overlap in terms of the susceptibility of some of the neurons in the brain. And so this is a monkey that was exposed to lead as an infant on the right, a monkey that was not exposed to lead as an infant on the left. And the monkey on the right, that is Alzheimer's pathology staining. That's a tau, neurofibrillary tangles, one of the hallmarks of Alzheimer's disease. And so exposure as an infant led to late life neurodegenerative changes. Here's another example. This is microglial priming. So this is an animal that was exposed to a bacterial wall, bacterial membrane component on day 5 of life, and then as an adult was exposed to rotenone. And on the far right, if this animal was exposed to this, basically an infection, as an infant, as an adult, it was much more susceptible to the toxicity from a small dose of rotenone. So it's microglial priming. Microglia are the immune cells in the brain, and they become overly aggressive if they're exposed young to this bacterial product. Here's another one. This is a combination of, so this is being exposed to these pesticides as an infant mouse, and again as an adult. And it shows counts of dopamine neurons. It's kind of hard to interpret, it's a lot of information, but the same point. If this animal is exposed as an infant and then re-exposed as an adult, it's much more toxic than if it had not been exposed as an infant. So the same adult dose is much more toxic. All right, finally, what are we going to do about this? Well, we can ban dangerous pesticides like most of the world has banned paraquat. We can ban TCE, and we can clean it up, and we can reduce air pollution, and we have in the United States. Look at Los Angeles since the 1970s. Incredible improvements there.
But don't forget the last one. Fund epidemiology.
Fund episode. Good luck with that. Yeah. Don't just ban the NIH and the CDC and the EPA and NIOSH and physical activity. Why not? Right? It makes you healthier in every respect, and it probably slows Parkinson's. Diet, Mediterranean diet, personally, I believe has to be eaten in the Mediterranean. It's definitely more effective there, but fresh fruits and vegetables, reduce animal fats. And caffeine is protective in epidemiologic studies, so enjoy your coffee. Reduce exposures, buy organic produce. It's more expensive, but you really need to do that. And organic meats, whatever that means. But animals that are not grown in a healthy environment are not healthy food. Know what's in your water. I know in San Francisco we get, at least annually, maybe quarterly, we get reports on the contaminants in the water. And I know in the Bay Area all the water supplies are pretty good. But a lot of people in this world still drink well water, and they certainly better know what they're drinking. And as far as reducing exposures in your home, the Mountain View example of all the Superfund sites is a great example. That's there. We know about them because Lenny Siegel looked for them. So if you don't look, if your community doesn't look, you don't know the history of what's under your home, that doesn't mean that it's not there, right? If we don't look, we don't know. Those pesky government regulations. So some of you may be aware TCE was to be banned in this document on the left. I think it was from December. TCE was to be banned, and that on the right, that ban was stayed by the current administration, and the courts are giving the ban, or the stay on the ban, at least until around now. I don't know the status of this. There are a lot of environmental groups that are suing on behalf of supporting the original EPA ban that the Biden administration put forward. I'm going to stop and take your questions.
Okay, can you guys hear me? Okay, great. Well, thank you so much for that great talk. That was really wonderful. It was really nice to see the history and some of the examples of the twin studies and the evolution of the work that you're doing. So thank you. I think my first question is, how do we find out if we're sitting on TCE exposure in Berkeley? Is there a way? Is there a local resource?
There are. I don't know about Berkeley per se. Yeah, I would. Berkeley's rather progressive community.
Exactly. Yeah.
And so I'm sure there are resources here. But know the history. I think you can rely on government to do this, but also know where you live. What was there a dry clean? A lot of these dry cleaning operations were just little mom and pop storefronts. And back in the 50s and 60s, they would just chuck that stuff out back. And so, know the history of your property. I think is important. But I don't know any specific.
Well, I know on the East Coast you can test for radon under your house, but I guess that's probably not common to test for other things. So.
There are certainly commercial services that will come out and measure TC in your house, but I think they're fairly expensive. It's not sort of you can just buy some little device and.
Yeah. Well, we'll open it up for questions. Let's make sure you have questions. I'll bring the mic back.
I'll start here.
Oh, sorry. Yeah, go ahead.
I'm never sure whether all the things I hear about that increase the risk of getting Parkinson's have anything to do with what you already have. So all the things that are listed as increasing the risk of getting Parkinson's, once we already have it, do those things have any effect?
Excellent question. We don't know very much about environmental impacts on the course of the illness. So we looked at Camp Lejeune, and we think that maybe people who are exposed to TCE have a faster rate of progression, but we don't know. Usually once people have Parkinson's disease, they become very careful about, they're less likely to be exposed, I think. Partly it's a function of the illness is that people are less likely to be out in the field applying pesticides, things like that. But we really don't know much about how those risk factors affect the course of Parkinson's. It's a great question.
What counts as a head injury?
What
counts as a head injury?
Oh, what counts as a head injury?
Yeah.
That's also a really great question. Most of the studies that have been done consider a loss of consciousness or amnesia. but that's actually a very tricky question, because if you do studies, say with some fancy imaging or a PET scan that measures brain metabolism, sometimes what appears to be a very mild head injury actually has a very significant impact on brain function. You know, we're seeing it now with all the CTE, chronic traumatic encephalopathy, in former football players. And you can see in autopsies of people who were in high school, playing high school, we do see sort of a dose response that you would expect. So it's worse if you play professional football, compared to college, compared to high school. But even relatively mild injuries can have a big effect on brain function. So it's hard to say.
Hi, thank you so much for what I would call a deep dive into the environmental influence on Parkinson. I just had a question about vinyl chloride. So PCE and TCE, what is vinyl chloride and how is it related risk-wise?
Vinyl chloride is actually one of the breakdown products in nature of those. So there's no epidemiology whatsoever on vinyl chloride in Parkinson's. Vinyl chloride is, as I understand it, is largely used sort of as an industrial intermediary type compound now, so producing other things, producing plastics, of course.
Polyvinyl chloride, for
Yes.
instance?
But there's literally zero human epidemiology looking at possible risks.
Oh, okay. My father worked in a PVC manufacturing facility in Louisville, Kentucky, most of his adult life, and it does cause liver cancer. There was a big deal about that, but I was just wondering about my own exposure. And so there's no studies basically.
There's no studies that I'm aware of.
Yeah. Okay.
Looking at vinyl chloride. Yeah.
Thanks very much. I know you stare at numbers all day long, and it's really helpful for us to have your takeaways on all that.
It's much better to stare at people. It's much better to stare at people than
Here's a question for you, Dr. Goldman.
Hi. So I was the one at Camp Lejeune, and so it's considered by VA as presumptive source of my Parkinson's. But that makes me wonder what genetic component there might have been as well. And I have 4 siblings, none of whom have any of these issues. But I also have 3 children, and if there's a genetic thing, I'd be interested to find out for their benefit.
Yeah,
Because if they
well-
need to start, if they're already in their 40s, they need to start modifying stuff if there is a genetic component in the situation.
Well, again, think about those causal pies that I showed. It's, it's some of this, some of this, some of this, some of this, put it all together, and it's enough to cause Parkinson's. But and most of the genetic factors that have been identified, the 90 SNPs we call them, by themselves increase risk just a tiny amount. And so you don't necessarily have much of a genetic risk factor. But nowadays, I know more and more people are getting genetic testing done. I don't know if that's, many people probably had it. I don't know how routine it is in clinical practice, but if you're concerned, you can certainly get a genetic panel like that. Generation Studies offering that. But if you get your genetics checked, once you know it, you can't unknow it. You know? Like, so I did 23andMe way back in the day when they were first starting out. We were working with them to develop their PD-specific messaging. I chose to not look at my LRRK2 gene because I was afraid. But but if once you know it, you can't unknow it. So think about that.
Okay, we'll take
I'm
a
not-
question over here.
I'm gonna follow the microphone here, so.
Good morning, thank you. I'm relatively new to all of this because I was just diagnosed on July 10th. And probably like most people getting such a diagnosis, you spend some time talking to Dr. Webb. And one of the first things I ran across was a new survey, a new study out of England indicating that a virus may trigger Parkinson's disease. So if there's my hereditary stuff, which I have no control over, if there's the environmental, which I have maybe some control over, and there's a virus in the air, what does one do not to get Parkinson's?
I think you're in good hands here with Dr. Goldman, because he's just putting in a grant to
Thank
study
you for
this.
asking that. How can you be on the grant review committee? So we did just 2 days ago to submit a grant to study that very specifically, to look at potential viral causes of Parkinson's. It's been, that idea has kind of floated around for a while, but there hasn't been good data, partly because, just like anything else, it's really hard to measure if someone got virus X 40 years ago. But there is a little bit of data suggesting that influenza A could play a role. There's also herpes simplex type 1, which most of us have had. That's the sort of cold sore one, but most people who are infected don't get cold sores. And other viruses, so that's great. And you know, there's a little bit of data that immunization, sorry, RFK, might actually be protective, but it has been not sufficiently studied, and we're hoping to get funding to do that. We want to study it in 3 settings: the human epidemiology. We want to try to make those associations. We also are going to test combinations of viruses and toxicants, paraquat TCE, in animal models and also in cellular models. Hopefully we'll know more in a few years about that.
My grandfather died in 1962 and Parkinson's disease. He was, before that, he was known to have been exposed to lead, probably even worse. That was at the DuPont Corporation, where he was mixing gasoline with liquid lead. And, so you just imagine how terrible that is.
Yeah. Well, our kids have got way less lead exposure than we have. Generationally, people born in the 50s and 60s had incredibly high lead levels that today would land a kid in the hospital for lead removal treatments. And also, speaking for my generation, we're less intelligent than we would be otherwise had we not been exposed to lead, and primarily lead in gasoline, which we all breathe. And so the good news is that the average lead level was probably 15 to 20 in the 60s, and now it's maybe 1. So our kids and grandkids are going to be smarter and are going to have healthier brains.
Yeah, and luckily we have unleaded gasoline as well. My first question back here.
Thank you very much for your lecture. I want to congratulate you on keeping me awake for an entire statistical outlook at what's going on with Parkinson's. My concern is the current administration and threatening research across the board. Is there any movement within the PD kind of science that is proactively looking at what this might mean?
Well, the entire research community is affected, not just by what's already happened, but by the uncertainty. NIH has been the main source of funding for Parkinson's research, and there's no telling what is going to happen to the NIH. So, we're all talking, and this grant that we just put in was not to NIH. So it's, unfortunately, we're all chasing the same dwindling pot of money, and the foundational support is more important than ever. Fox Foundation, Parkinson's Association, all the foundational funders of research are getting a lot more pressure on them from the research community. But for sure, we're all talking about it, and everyone's afraid that their funding's going to get pulled. It's going to have a terrible chilling effect on research. Hopefully it's temporary, but tearing down an institution is a lot easier than building it back. So these impacts could last for a while, I'm afraid.
Just to follow up on that, I'll send out an email to everybody who participated and give a link to ways that you can get involved in that, express your opinion about closing of these federal institutions. And also there's an organization called PD Avengers that I follow that does a lot of activity around banning pesticides. So I'll send that link out as well. I think we have a question in the back.
Thank you. I've really enjoyed your talk. I'm a retired public health scientist, and times like this, when I see this kind of data, I wish, well, maybe I wasn't retired. But I had the opportunity to attend a medical appointment with a family member that has Parkinson's disease, and because of my background, I asked about environmental factors, and she said, no, it's all genetic. And then about 15 minutes later, talking to the doctor, she says, oh, well, Agent Orange could cause Parkinson's disease. So, I knew a little bit more than that at the time, but I'm curious, two things. One is, how and why do you think someone that is a neurologist wouldn't know a little bit more about this, and what's being done on that in that regard, or what's being published that, in neurological journals or whatever? Yeah. And also kind of curious separately about, you said you thought there was an increased rate of Parkinson's disease. I'm just curious what you think the factors might be related to that.
Yeah, well, we know that something like 70% of doctors don't take an occupational history, primary care doctors. So you think about your potential for environmental exposures, a lot of it is through your work, and docs don't even ask. You spend a third of your life at work. Well, the field in general has been very genetically focused forever, because the genetics tools are perceived as being more definitive, I think. You measure this, you test for the genetic variant, and it's either there or it isn't. In epidemiology, our work is dirty, it's imprecise, it's hard to replicate, it's slow, it's not sexy at all. It's not what I got it. And the genetics, it's sexy. It's technology. It's these fancy tools, these fancy machines. A lot of people people are making money from it. Nobody's making money from human epidemiology. And so the word it just isn't there in the community. And every time they find these genetic studies get bigger and bigger and bigger and bigger, and they just never find any more genetic associations. This 25% is all we can explain with genes. And I don't know why this person isn't. Yeah, it's sad, but I'm sure it's the norm, not the outlier. I'm sorry.
No, that's what I was afraid of. But as far as the increase,
Oh, the increase, I think it's industrialization. I think it tracks with industrialization. You are what you breathe, drink, eat.
Speaking of eating, you said to eat organic. Do you mean like the Dirty Dozen, or do you mean like everything? Like all veggies and fruits and things?
Dirty dozen, you mean like strawberries and
Yeah, like the list they say to eat organic, or do you mean like everything?
Don't eat.
Or the Dirty Dozen, sorry, I mean like
Yeah,
do you mean
yeah.
like the list of things to eat organic or don't eat organic, or do you mean like just eat everything
Well,
organic?
you should eat well, and if you can afford to eat organic well, that's better.
Okay.
But it's like don't avoid eating vegetables. If you're what are you going to replace them? What are you going to replace them with? So I think organic, assuming it's California organic is better than national organic. I'm not really that familiar with the regulation of things being called organic, but you should try to not eat pesticides. You should try to not eat heavy metals. So when you're eating fish,
What's an herbicide then? What's an
herbicide kills weeds, kills plants.
And there's no studies on herbicides?
Paraquat is an herbicide. Glyphosate is an herbicide. 2,4-D, Agent Orange is an herbicide cocktail. There are lots of herbicides, hundreds.
Pesticides and herbicides.
We loop them all into the same pot. We call them pesticides. So pesticides means insecticides, herbicides, fungicides, it kills mold, mildew, and even rodenticides, it kills rats.
Could you talk a little about dairy?
Wait, we have a question in the back, and then we'll come back to you, Karen.
Is anybody studying medical chemicals? All the stuff that doctors use, all the different chemicals,
Yeah.
and then when we have operations, we have all kinds of chemicals that we're exposed to. Is anybody studying that?
Yeah, actually, it's a bit easier to study that because it's in the medical record, and so we can kind of extract that data. And that's been looked at both for things that increase risk, but also for things that decrease risk. So there's a lot of focus on what's called repurposing of medications that are used to treat something else, say a treatment for rheumatoid arthritis that reduces inflammation. Maybe it's effective for Parkinson's. So there is a lot of work on that, and I'm not familiar with much, if anything, about medications and drugs that would increase risk. It's a good question. There's a lot of work in that area.
We can be confident that there's not a lot of TCE in hospitals for cleaning up after.
Not anymore, but there probably was, and maybe still in the soil. That's, ethylene oxide is the main thing that's used for sterilization, and there is some. I've seen a little bit of research looking at that as a possible risk factor, but it's not very convincing. As for things like anesthetics, there has been some work. So TCE was used as an anesthetic. And there's a little bit of data out there, but it's not very strong and convincing about anesthesiologists who, of course, would get exposed over a lifetime to various things in small levels, about them maybe having a higher risk, but that data is not strong at all.
Yeah, I had a question about dairy. Is dairy considered a risk factor because of vitality, or is it for other reasons? And
Yeah.
can you talk-
There are some studies that
Maybe you can repeat that, Dr. Goldman.
Oh, yeah, it's a question about consumption of dairy products. Might that be a risk factor because of cow's diet? Well, there have been some studies that found an increased risk of Parkinson's disease associated with more dairy product consumption. And the hypothesis behind that is that toxicants concentrate in fatty things, and dairy is very fatty. And so maybe it's people who eat a lot of dairy are getting a lot of fat soluble toxins. Another reason to eat organic, right? The cow, you don't want the cow to be eating pesticides or getting treated with hormones or antibiotics or things that.
So because of that problem, you still have a problem with dairy. For Parkinson's.
Well, we don't, that's a good question. We don't know whether clean dairy is safe. But my feeling is that any association with dairy is related to the toxicants in dairy. I can't prove that, but that's what I think is going on there.
It's me. Hey, thanks for a great talk. The mitochondrial poison was a really interesting concept, and since that's kind of like the powerhouse of the cell as I've read it, I'm curious, there's a lot of products that claim to sort of help with that, like red light therapy or PEMF. Any insights or thoughts about that?
Oh, yes.
Yeah, there were some studies. Coenzyme Q was one that was thought to, there was actually a clinical trial. I'm not really up on some of those, but it was not a successful trial to try to increase mitochondrial health and function. I'm like a skeptic first. That's sort of the way I view life. So I want to see data before I personally would feel like taking some sort of supplement or something like that. We don't, there's basically, I'm not aware of anything, unfortunately, currently, anything that is a disease-modifying therapeutic that exists. Though you guys are probably, many of you are probably involved in clinical trials, and there are so many products in development and taking lots of different approaches, but certainly improving mitochondrial function is a rational target for a therapeutic based on what we know about the mechanisms of these environmental toxins. Yeah, for sure.
We have a question here.
You showed a chart with the relationship between linking paraquat and I think it was the GST gene. How about the LRRK2 gene? Will that be comparable?
That's a great question, but the LRRK, that's really hard to study. LRRK2, there's a handful of different genetic variants in this gene. The most common one is called the G2019S variant. It's rare. It's not, among Parkinson's related genes, it's common, but in the population, it's very rare. So to do a study like that is extremely hard. But we have actually looked in the PPMI study, I don't know, some of you may actually be involved in that. There are a lot of people with genetic variants in that study because they've been recruited. And so we were able to look at the interaction of pesticides and solvents with in people who have who carry that gene. Well, people with the LRRK2 gene, only about 30% of them get Parkinson's disease, which is a lot, but 70% don't. So we look in people who carry the variant, and we try to see what's different about people who do get Parkinson's with the variant and people who don't get Parkinson's with the variant. And we did find an association with, not with LARC, but with the GBA genetic variant, where people who were exposed and had the variant were more likely to develop Parkinson's. Yeah, but a lot of these, to look for interactions between different things, just statistical level, you have to have enormous study populations to test those sorts of things.
Here you go, Dr. Goldman. Here you go. Hold it right up close to you.
Oh, okay. Yes, I spent 20 years working in a building that I didn't know was toxic until we got moved out and they were going to tear it down and build new. And then they had to stop because underneath the sidewalk there were just all kinds of toxic chemicals and bad stuff. And so I'm wondering, well, I have a feeling that affected me and maybe had something to do with my getting PD.
It's hard to know these things.
Advice?
Right? We're walking causal pies, and right. So if you take away a slice of that pie, would you have had Parkinson's?
Maybe not.
We can't know that, right? We don't. It's just
Do you know
impossible.
what types of
we know
toxins?
that.
Yeah, the state sent me letters telling me.
Do you know what chemicals?
Right offhand, no.
You know, a lot of these things are relatively easy to clean up. Some of them aren't. Things like heavy metals, you gotta remove the soil and a lot of it and put it somewhere in a toxic waste dump forever. But something like TCE in the groundwater, they're pretty reasonable ways of reducing those levels. Basically, you put a couple of vents down in there and you just have a fan sucking out the fumes and goes up into the atmosphere. So they're...
Cal OSHA came and did that
Yeah.
for a week. They had all my things that were there. I had to throw them out, and resources for teachers. Because that where the storage for resources for me was
It's hard, but it's important to know.
Black
history.
mold. There was black mold all over it.
It's worth knowing what's underneath where you live.
There's also ways to have your own profiles checked. Like there are certain naturopaths that do hair analysis that look at chemicals in your hair. I don't know, not necessarily. He's skeptical. But you can. I mean, for example, mercury. You can look at what your mercury levels are, and then you can just reduce your tuna consumption. I mean, there are small ways that you can.
I would just say reduce your tuna consumption. Don't get the test.
Reduce your what consumption?
Tuna, mercury in tuna.
Oh, okay.
Tuna, like tuna fish,
Yeah.
tuna.
A lot of these tests I don't trust. I think they're more about getting some money for the lab and the person doing the testing. We've all got stuff in our bodies, and reduce your exposures. That's my advice. Whatever level you have in your body right now of mercury or cadmium or lead, that's too bad. Do what you can to reduce it. So don't eat fish that are at the top of the food chain, right? Eat sardines, eat little fish, and eat healthfully.
So I have a quick follow-on question to the exposure. The one slide that really caught my attention was the prevalence of Superfund sites here in our own peninsula, for example, and then the insidious notion that TCE can travel through the soil and send up fumes into our own homes. Please tell us something's being done to clean up those Superfund sites.
The Superfund sites, they are, yeah, they get cleaned up. But your point, are they continuing to track the plumes? The plumes travel, they don't travel, fast, but they travel. But the biggest issue is that we don't know they're there. I was just asked to help a housing group in San Francisco in the outer Sunset, where they discovered a plume because there was an old dry cleaning site and they wanted to build housing on top of it, and they hadn't tested soil before they could build, and they discovered it. And the neighborhood was very concerned about that. But if you look, you'll find it. And so Mountain View, they found it, and then they can clean it up. They can do something about it at that point. But if you don't know it's there, that's the bigger problem.
So typical soil studies will find THC.
Yes.
TC rather.
THC, that is. It doesn't travel as far.
Still living last weekend.
Can I get a question in here? I wanted to rewind to alpha-synuclein. Given all the indications and the different kinds of evidence pointing towards mitochondrial dysfunction, is it at least like a downstream thing of Parkinson's, if it's not the beginning of Parkinson's? What is the normal function of alpha-synuclein, and does it link to mitochondrial function?
There's been tons of work trying to figure out what alpha-synuclein does. And it seems to be important in vesicle mobility and vesicle. So vesicles are little bubbles in the ends of neurons that carry chemicals, neurotransmitters, things like dopamine. And they have to fuse with the membrane in order to release the chemical out into the synapse for it to do its function. And alpha-synuclein seems to be involved in the fusing of vesicles to the membrane to allow the release of this. Now that's part of the issue with developing drugs, so there have been lots of efforts to try to, we know that when the synuclein, certain forms of this protein can aggregate and clump and form Lewy bodies in the neurons. And there have been a lot of drugs and antibody studies, some of you probably know those, to try to reduce levels of alpha-synuclein. And the problem is, of course, this is a protein that's got some, important function in the brain. And so there's these what are called off-target effects when you try to develop a drug against something that has an important function. You've got to be careful in how you lower it because it's got a role. I don't know what the relationship is with alpha-synuclein and mitochondria per se.
Perhaps there's not one, which might be a really interesting angle.
Dopamine neurons are, they kind of live on the edge. They're metabolically really very active. They've got a lot of iron in them. That's part of the brain that's most affected. It's called the substantia nigra, which means black substance, and it's black. When you look at a brain, you can see it's this little black area. It's black because of neuromelanin, which binds a lot of iron. It's a breakdown product of dopamine, and it binds iron. And iron is very reactive. It rusts, right? It's an oxidant, pro-oxidant. And where am I going? So dopamine neurons, they've got a lot of this iron, and they're also very metabolically active, so they're super dependent on their mitochondria, and they're under oxidative stress, so they're neurons that are like living on the edge. And so they're kind of more susceptible than other neurons to injury.
Oh, hi. Thank you so much for the presentation. I have two questions. One is, you mentioned the vapors from the chemical TCE. Would air purifiers help if we can't identify the vapors, and do air purifiers work in the house?
I mean, typical ones probably wouldn't do much, and most of them are particulate, they filter out particulate matter, which is probably good too, right? Because particulate, especially the super small ones, are bad for the brain probably. As far as filtering out TCE, I doubt it. I don't actually know. Maybe, you know, activated charcoal, and I should know this, I'll go look it up later. Perhaps there are some that could be effective. But more effective would be knowing, you know, if it's there.
Exactly. My other question is a little bit more specific. It has to do with jet propulsion fuel. I'm wondering if you know of any studies that have shown the link between that and Parkinson's.
We're actually currently looking at that in a study in a VA cohort. I'm not aware of any associations, so JP4, JPA, they're military jet fuels in particular that we're looking at, but they're all kind of related. The problem is it's very, people who work with those fuels also work with TCE, work with other solvents, so it's hard to sort of pin it down on any particular agent. There's not strong data implicating those, I can tell you that. We're not currently seeing anything in our study.
Thank you for that question. Is there something that sparked that particular question for you?
My husband was served in the Navy on board ship, and he has Parkinson's.
Mm.
And he's exhibited prodromal symptoms for a long time before he was diagnosed.
Well, thank you for all of those of you who have shared your personal experiences today. That's really nice. I think we'll take two more questions. We have Norm here who has a question, and then we'll go over to the other side, and then we'll wrap up so you guys can get on with your day.
So I know my genetic factors are
Close to
involved in my Parkinson's, and I have a pretty good idea of what environmental factors influence it also. Do you have any tips as to how I explain to my children the potential risks that they face?
Great question.
You repeat it?
Yeah, okay, you good?
You were concerned about how you explain the risk to your children. In general, risk to first degree relatives, parent, child, sibling, is low. If we didn't know anything about your genetics or your exposures, just taking all families, people who have an affected first degree relative probably have about twice the risk of getting Parkinson's compared to people who don't have an affected first degree relative. But most people with Parkinson's don't have any affected relatives. Most, we call it sporadic. Probably not sporadic, we just don't know what's going on. But in general, the risk to your children is higher than if you didn't have Parkinson's, but it's not high. If you figure your lifetime risk, in the general population, the lifetime risk of Parkinson's is 2%, maybe, you know, lower in women, a little higher in men. Twice that is still pretty low risk. You know, of course, their particular risk is going to be, if you have genetic factors, then that would potentially impact their risk. Some of these things, you know, they inherit a copy of a gene from you, a copy from their mother, and so depending on the gene, it would impact their risk differently. But as I said, even people who have a genetic form, except for some of these extremely rare ones, like in that Contourzi kindred, even the Parkinson's genes, most people who have those Parkinson's-related genes like LRRK2 or GB1, most of them don't get Parkinson's. So just in general, I would say, the risk is slightly higher than the general population, but it's still not high. It's less than diabetes. If you had type 2 diabetes, their risk would be higher than considerably compared to Parkinson's.
Our last question here from
Yeah, I'm hard of hearing, so I may have missed this, but to tell a little bit more about race, ethnicity, men versus women, where they live, other factors that might affect Parkinson's.
Good question. So I said that men are more frequently affected than women. That's not entirely true. In Japan, women are more frequently affected than men. In Japan, women historically have done most of the farming. And everywhere else, historically it's men. But we're not really sure what makes for the sex differences. It may be exposure related, it may be something, there's a fair amount of data that estrogen might be protective against a range of neurological insults. But that's still a big question. Geographic variability and racial variability, first of all, we're not absolutely sure it's real. But there's a fair amount of evidence that rates of Parkinson's are lower in Africa and are lower in African Americans. Again, with a caveat that may be our inability to find everybody, right? They don't go to the doctor, they don't go to a good doctor, they don't get diagnosed. But that aside, yeah, there may be racial differences. And one hypothesis is that melanin, so Africans are more heavily pigmented than Caucasians, that melanin in the skin might bind some of the toxicants and thereby prevent them from getting into the brain. But we really don't know. As far as geographic variability, there's definitely geographic variability. Just in the US, the rates of Parkinson's are widely different around the country. Again, I hate saying this, we don't know for sure if that's real. Because people with Parkinson's are gonna go, they might spend their whole life, here in one place, and then they move to another place because that's where the good medical care is. Or that's, they're gonna find more Parkinson's disease in close proximity to medical centers where there are Parkinson's experts. And so that's, it's hard to study, but we do think that it seems to correlate with industrialization. And that may be why rates are lower in Africa, because it's later to industrialize. And pollution is lower there over the past 100 years.
All right, well, on that note, we're going to say thank you so much, Dr. Goldman, for joining us today. This was really fascinating talk.